We will now discuss the two types of questions that you need to be solving for the IR spectra and from the IR spectra. You might be given a chemical structure and you might be asked its corresponding IR spectra. Or you might be given a spectra and you would need to solve for its probable chemical structure. I have listed out the sequence that you should follow when you're solving from both of these questions. For you to be able to solve IR questions, one of the first things that you should be familiar with are the functional groups. At the end of this document labeled IR examples, I have listed all the functional groups that you are required to know for this 20 to 30 lab course. Their general structure and an example of an actual structure. You should be familiar with alkanes, alkenes, alkynes, terminal as well as internal alkynes. Internal alkynes and nitriles. Both of these share a triple bond. The alcohols have an OH, whereas an ether has an OR or an OC bond between it. So this pattern of the atoms or the arrangement of these atoms on the chemical structure, you all should be familiar with the functional groups. Primary amine, secondary amine, tertiary amines. As the name indicates, tertiary amines has 3 carbons around it. Secondary amine has 2 carbons on it, while a primary amine only has 1 carbon. Remember, carbon and nitrogen, they are multivalent atoms, and hence they can individually have their own primary and secondary designations. On this particular example that I am showing you here, if this is the arrangement of electrons and atoms, if we look at this particular molecule and we have this particular arrangement of atoms around carbons, we see that this particular carbon has 4 bonds on it, and one of the bonds is attached to 1 carbon. This carbon is a primary carbon, while the carbon which is surrounded by 2 carbons is labeled as a secondary carbon, whereas a carbon surrounded by 3 carbons is labeled as a tertiary carbon. And any monovalent atom on these carbons is labeled according to that. For example, if I have a structure like this particular carbon I know is a secondary carbon. If I have a monovalent halogen on it, I will label this as a secondary halide. Or I might have an OH on this secondary carbon. At that point, this molecule will be known as a secondary alcohol. So depending on which type of carbon the monovalent group is attached, you will label that as such. Similar to this situation, we have nitrogens. In this particular system, the multivalent nitrogen is connected to only one carbon, and hence this is known as a primary amine. The amine takes preference over a simple alkene. A primary amine has two hydrogens on it, whereas a secondary amine will have two carbons on it and only one hydrogen on it. What about a tertiary amine? A tertiary amine has no hydrogen on it. Now, this is something that you have to understand and remember. The tertiary amines are not visible on your IR spectra also. Why? If you remember, on your IR, we will only have on the first and the second region compounds with hydrogen on them. If they are electronegative, they will be a little further left. If they just have carbons on it, it will then be a little to the right. Since a tertiary amine has no hydrogen on it, you can differentiate between a primary, secondary, and tertiary amine. Tertiary amines will not be seen on the IR for region 1. The rest of the regions for the tertiary amine, a secondary amine, and a primary amine in these examples shown here will be exactly the same. Why? If I list out all my bond types, these are the only bond types that I observe besides the NH. Tertiary amine misses these NH kind of bond types. The C single bond N belong to region 5 and are not on our list that we can distinguish them from the rest. sp3ch will be seen on your second region. sp3 carbon are also in region 5 and hence not to be distinguished, not to be cared for. But understand how we know what is a primary, secondary, and tertiary amine. Moving forward, a ketone will have a setup of carbon, carbonyl, and another carbon. Whereas an aldehyde will have a carbon on the carbonyl and a hydrogen on the other end. A carboxylic acid has a OH instead of this H, while an ester has a OR instead of this H. An amide is a carbonyl next to an amine. If both these groups are hydrogens, or both these groups are carbons, or if one of them is at least a carbon, we still call it as an amide because of this particular setup, a CO next to the nitrogen. Let's start from our first type of questions. If you're given a chemical structure and you're required to guess out of the given few options, or if you are asked to draw its IR. And when we ask you to draw an IR, it is tentative. You do not have to draw to scale. But the first thing is to write out the structure. When I write out my structures, I write all the hydrogens. So I do not miss any of these on my chemical structure. This is especially useful when you assume that you know all your hydrogens and bonds, especially in the beginning. Now let's list out all the bond types present in this molecule. And it does not matter what is the order that you're choosing. I will start from the right hand corner. I have a OH. I have a C single bond O. I also have a carbon hydrogen bond. Now I need to be specific in mentioning that I have a sp3 carbon hydrogen bond. I also have a C double bonded oxygen bond. This carbon, which is sp3, and this carbon, which is sp2. I can write it. However, when we were looking at our IR packet, how to look at the IR packet, I specifically mentioned that all the single bonds except for C double bond O are useless for our IR. But nonetheless, I'm going to write it for this particular example. So I wrote the sp2 carbon and sp3 carbon bond also. I've already listed the sp3 carbon hydrogen. Just for the sake of writing all the bond types, I will write this bond type also. I have a sp2 and sp2 carbon here. Do I write that out? Absolutely. Except for all the single bonds, we should be looking at all the other bond types. If you think you're done, look again. There is one more bond type that we forgot to write, and that's where writing out all your hydrogens on your structure will help you. This is a sp2 carbon hydrogen bond type. sp2 carbon hydrogen bond type, sp2 carbon hydrogen bond type. Once you have listed all the bond types, your next assignment or next step is to find where these bond types lie. And this is where your packet is going to help you. If you look at the first page of your IR packet and you compare where is the OH, which region the OH belongs. And we see that the OH belongs to 3200 and 3500 centimeter inverse region, region 1, and it is a broad, round peak. In my list, I will go ahead and record this information. OH is in region 1 and it is a round, broad peak. What about C single bond O? The C single bond O peak appears in region 5 and it is usually a strong peak or more distinguishing peak. So C single bond O is in region 5, strong peak. What about sp3CH? sp3CH is in region 2 and it is a multi-pointed strong peak. Region 2, multi-pointed strong peak. C double bond O is in region 4, strong peak. Region 4, strong peak. I'm not going to worry about these two single bonds. We will only worry about the hydrogens and the single bond and the C single bond O. Where does the sp2 carbon bond peaks lie? They also lie in the second region, but their shape is a little different from sp3 carbon hydrogen. It is on a left shoulder and usually not that strong a peak. So this also lies in region 2, left shoulder of sp3CH. And the last one